Okay, so exam, we started a little bit about it, micro, 7 and 13 in here. So we have how many on? 4, is that right? Okay. So the learning curve is starting. So with exams, first exam, what to expect, now you know. Quiz will go up next time, just like this time. And hopefully it won't be a Labor Day weekend in front of it. And you'll have time to go through it. But the suggestion yesterday for Preston, he wasn't in micro, was that take the quiz and study at the same time. So take your time. There's no time limit on the quiz, and work through the questions as you study. And maybe that'll help put it together. But the quizzes and the review questions were pretty important. At the end of each chapter, those questions were in there. The quiz questions were in there. And that was pretty much it. So the pool was somewhere in the range of 40 to 60 times 4, 250 say questions, around that, 240, 250. And you had 50 questions come out of that. So you had, if you only did the quiz one time and you didn't really look in the back of the chapters, do those questions too, then you were flying without, some help. So the help is there so you get used to studying questions and see how the material is presented so you can recognize the answer. So you're kind of working toward a final goal of the program is not only, we're pretty good about getting you a job, okay? You graduate, that's the other goal, right? That you graduate and finish, and then you get a job. So you be proud of, you went to college and you have a job waiting on you when you graduate. How disappointing if you got out of college and there's no job, right? That happens. I mean, there's degrees on campus that they get out of school and graduate and there's nothing for them to go to. And they kind of look back and go, maybe I picked the wrong degree. That's one thing we can tell you, that there's jobs, okay? We get daily updates of, hey, there's somebody else looking. There's a lab manager position in Arkadelphia today, open. There's a lab manager position in DeWitt Hospital, open. So they're not only are you getting a chance to graduate, I mean, you want to jump to a seat that pays more, those are open. So, I mean, you could, parlay that into something really good. The other point is passing the board. The board is our way of gauging how well you're retaining, how well you're prepared to pass a evaluation of your knowledge. And that has been taken lightly in the past. They didn't really harp on that a lot during orientation because they didn't give me that part. But in the past, it has been an ongoing issue because it's not a requirement. Some places require it, some people don't, some people just suggest it, but it sets you up for everything else to come. We had to turn people down for Miss Carly Farmer's position because they weren't board certified. There were past graduates that wanted to come back and be in that role here. And one of the requirements was they had to pass their board, and some were still working 5 years away from graduation without their board. So we know that's an issue. But the way we're testing you is to help you pass the board too. So multiple choice for me, you're going to have a multiple choice board. The information, the lecture, you like the lecture. Well, the lecture is going to go for your work daily. You're going to remember our lecture. You're going to remember our talks. You're going to remember the stories I told you, right? Those aren't on the board. So the test that we give you is to prep you for the board, to show that you can pick out the right thing. What's that?
So can't you get paid more?
Oh yeah, yeah. Always ask for more pay. Yeah.
Why wouldn't you take it?
We know all that we know all the reasons you should.
Yeah.
There's still graduates that don't feel the need to do it. So we know that. We know that if you wanted to work in Memphis, which is just down the road, big, St. Jude's sitting there, AEL sitting there, you have to have your registry passed. You have to have your boards passed. Okay, Tennessee does not give you a free ride. They have a license that requires the board to work. Okay, Louisiana, maybe New Orleans is where you always wanted to live once you graduate. You can't work in New Orleans with this degree if you don't have a board. California, love California, right? Have you ever been out there? San Diego up to San Francisco, great place, high wages, you know, fun. It's burning right now, but other than that, maybe you want to go there. You have to have a board. Okay, New York, you got to have a board if you always want to live in the big city, the huge, best city, you know, the biggest city that I've ever been to, right? So those are the things that we're prepping you for. So don't just, you know, one test doesn't make the career. One test with a new professor doesn't make, oh my gosh, I'm going to do that every time. But learn from that first one and be better prepared for the second one. It's coming quick. It's not like we're going to be spending 3 or 4 weeks waiting on it. I want to say it's the 23rd. Okay, a couple of weeks away. All right, so it comes back pretty quick. Today's lecture is great information. It's the antibodies, so it's definitely important, and we're going to talk about how antibodies are structured, their types. So we're going to group them, what each group does, and so you get a lot of info that's going to come back to you. Each chapter we look at, this one sets it up. It was one I wanted in Unit 1, but we just didn't have the space and time, which is okay. We might find that out again later. We might be on a lecture that we need 2 days for. I don't know yet. We might get 50 minutes in and I go, I need another day. We need to finish this, say, complement. We need to spend a little more time there. So I don't know yet. It's the first time through this text with you. I've been through it many, many times, but with you, I don't know. So, and I don't want to get rushing and just to stay on schedule. We're doing that the best we can, but definitely don't want to. Or as the group, I don't know if y'all heard them in lab, they were asking about, have you given them the 9-unit test yet? I gave them a 9-chapter test last year.
You're not going to do that, right?
No, I'm not going to do that. But they were just like, that was way too much. They were like, we cannot get through this stuff. That was horrible. Thank you. No, you have another test added this year. They didn't have last year, so we can keep it to 4 to 5 units at a time. So 4 or 5 chapters. All right, so overview of today is tetrapeptide structure of the immunoglobulins. So if you're seeing this word for the first time, hopefully you're not, but hopefully you'll understand why we call IgG immunoglobulin G. Light and heavy chains, we'll talk about those pretty good. 3-dimensional structure. The characteristics of each antibody. Antibody diversity theories, we kind of lead that. It kind of shows you where we started with the theory of why we have a specific antibody for a specific antigen. And the genes on how that codes. I'll go ahead and tell you that this is tough to get through, genes coding for immunoglobulins, but if you went in and did handwritten tutorials the other day, he has a really good one on the gene coding for immunoglobulins. He actually draws it out and shows you the different genes and how they splice each other out to get to the unique immunoglobulin. So it's very good there for a review. If you get through with today and you're going, I had no idea what you're talking about. Go back and look at handwritten tutorials on YouTube. And then we'll finish up with monoclonal antibodies, which is the exciting part of the lecture today because this is going on today. We're using monoclonal antibodies to treat patients with, okay? Have a great story with my wife. She's been given a monoclonal antibody as a treatment for an autoimmune disorder. Worked, right? We had to go to the infusion center, but we'll tell that story at the end of the lecture today. So you'll know that we have firsthand knowledge of how monoclonal antibodies are used. So as you say, this is not algebra, right? We won't be talking about something you get to the end and go, why are we talking about something I'll never do again or never see again or anything? Hopefully you're seeing that with our classes, that each class we're going through, you're unique, and we're going to see it again. So don't get to that point of, I don't know why he lectured about that today because we'll never hear or see that again. That's not our case. And you wouldn't expect that from a professional program, would you? Antibodies are immunoglobulins. Okay, they're glycoproteins, so they're sugar proteins. You've got to know that. That's a question that comes out. You know, what is an immunoglobulin? It is a glycoprotein. It has a carboxyl end. It has an amine end. They're found in the blood, serum, 82 to 96% polypeptide, and 2 to 14% sugar. So here's your, your protein part. That's your sugar part. We have 5 major classes: IgG, IgM, IgA, IgD, IgE. You've already seen some snippets of G, M, and D. Now we're going to fill in some more detail on each one. And these are the key elements of the humoral immune response that we've already been tested over. So how did they get their name? They got their name because they did electrophoresis. Okay? Electrophoresis pattern is the separation of proteins out of the serum. And the immunoglobulin was primarily found in this peak of separation. Okay, so you got your albumin here, you got your alpha 1, 2s, and beta peaks, and you got your gamma peak there at the end. And most of our immunoglobulins are located there. So if we think about it, if we have abnormality with production or overproduction of immunoglobulins, what happens? This peak rises. Okay? So you can see an abnormal electrophoresis pattern, which, believe it or not, the board will put one on there for you. So we'll talk about this in lab when we get to it, and we have a review of abnormal electrophoresis patterns. We'll do an immunoelectrophoresis in lab, but it won't look like this. We're going to do a little different technique where we actually put the antigen and antibody separated, run a charge, and watch them migrate toward each other through the gel. Okay, so you can see the specificity of one antigen and one antibody. They move toward each other. This is not a specific antibody over here to the antigen, so it doesn't migrate there, so it doesn't do this, it just does that. So we'll see that pattern, so we'll get to see some of this migration. But that's why they call them gamma immunoglobulins, is because they are coming from there. So the gamma peak. So this is our basic structure. So we're going to analyze the basic structure of the antibody. And we have 2 heavy chains and 2 light chains. So it's a tetrapeptide, 4 basic chain polypeptides, so tetrapeptide. And hopefully you get the idea. They've labeled them really nice for you. So they've labeled the constant region. This would be 1 constant, and it ends here. Okay, constant 1. Then you have a constant chain here. Okay, so this is your heavy chains. They're the heavies, and these out here are the light chains. See, they're a lot shorter. But hopefully what you're picking up is that we have constant, which means that's the same for, let's say this is an IgG. This is all the same for every IgG, this structure. But this is the variables out here. So you have variable heavy, variable light, and you have 2 sides of that. Okay, so that's our basic structure. We always draw them in the Y's, and you'll see why in a little bit. But there's a lot of good detail in the book, okay, about why we get the names. And you might be asking, where did we get all these names? So who wants to take on the first naming? I like to do the first one. Why do we call them IgG, IgA, IgM, IgD, and IgE? Who has read ahead for today's lecture and prepped and has read the first page of the chapter and can tell me why they got those names?
do have a question though. On this,
Mhm.
is it the purple ones at the end and then the white ones that are not constant?
Purple one's on the end. The heavy chain is purple. The light
Like the
chain is
whole
white.
purple chain?
The whole purple chain is constant. I mean heavy. Sorry, heavy.
Even the VH?
Yep, that's heavy.
Okay.
All the purple's heavy. All the white is light.
So all the purple
But
is
if you
constant
cut, but
then?
if you cut off here, this is the variable. So the variable's only on the ends of each chain. Constant is this part of each chain.
So the constant is not the in purple ones?
Yeah, this is constant still. Here's constant 1, heavy. Here's constant 2, heavy. Here's constant 3, heavy. Here's constant light. Here's variable light. So what you're seeing in the structure is the very end of each immunoglobulin of the Fab region. Okay, so it's the upper. Each of this here, boom, 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 boom. Those 4 spots are variable for each antigen. The rest of it below those, the rest of the heavy chain, the rest of the light chain is constant for each class of immunoglobulin. Am I seeing that, or is this?
It makes sense now, but I was asking about the variables.
Yes.
Those are
Variable
not
heavy.
constant, right? The in purple ones up top.
You're saying that wrong still.
How?
You just said the variable up here is constant. And it's not. This is a separate area of the chain. Okay? Chain starts here.
I said except that one,
Oh.
except the VH on both sides.
Yeah.
Okay. Thank you.
All right, back to my original question. Since you guys aren't getting it. What's the question? What's the answer? Sorry. How do they get their names? Nobody. Okay, think about it. When you get the answer, we'll interrupt, come back.
They're Greek letters.
They're Greek letters, yes. That they are. Alpha, mu, gamma.
Does it have to do with oxidation complement?
Oxidation complement. No. I'm not, that's too specific. I'm just looking for a basic answer. So somewhere on page 62, those that have their book. Okay, just leave it there. We'll keep going. All right.
The domains of amino acids.
Yes.
Oh, okay.
Domain, she said domains of the amino acids, right? So it's the structure of what? So the structure of the heavy chain, okay? So being abbreviations for immunology, the heavy chains are gamma, mu, alpha, delta, epsilon, respectively. Although each class has distinct properties, all immunoglobulins share many common features, so their basic structure is the same. So that heavy chain is what designates the name. So the change is going to be, there'll be a difference in the heavy chains of each one. FC, anybody know what FC means? These are all good things. I'm not giving you anything that's not highlighted in black in the chapter. Fraction crystallization fragment, and that is, if we were going back, which part of the immunoglobulin is the FC region? Connect that dot. Yes, so we consider this end, right, the bottom of the Y. We consider that the FC region. And that, you remember when we did the B cell, you remember the B cell had an IgM sitting on top of it and an IgD sitting over here on the other side of it when it was doing its development, when we're studying the development of the B cell. That FC region was binding to who? It was binding to cells, okay? So you always think about the FC region as being binding to the other cells, like the antigen presenting cell or the B cell or the macrophage, right? The antigen presenting macrophage. And then it gives that cell what? It gives it the antigen binding positions, which are the Fab region. What does Fab stand for?
Fragment antigen binding.
Fragment antigen binding. So it's a little more specific on where that name came from. But fraction crystallization is what? Is a technique that was used to see this, right? So you read the chapter, it talks about how the scientists discovered the immunoglobulins, everything from centrifuging them to treating them with different things and breaking them apart. Okay, so they had to like find them first. They found them, then they had to start taking them apart. And then the story finishes with, why are they different? How did they become different for each antigen? Okay, and that's kind of close to, not that far away from today. Okay, so we have found a lot of things from, say, 1950, 1960, right? So just in the last, what is that, 50, 70 years, under 100 years, we have everything that we're doing today, has been discovered since the 1950s. Okay, so this isn't like so long ago that we knew the structure of these immunoglobulins, but now we do. So you have to know the Fab region. Very important that you know that region is where the antigen binding happens, and the reason it gets its name. And the FC region, right, is where we bind to other cells. So we have the complement and FC receptor binding. So we see this word complement, and you're going to get that again. Complement comes back to coming up. We're going to talk about it, but it's a way of activating complement is that region. And also FC receptors are found on other cells. So we have the receptors on the membrane of other cells. They come together and bind up. Okay, attach, link. So the FC region has no antigen binding ability. It represents the carboxyl terminal halves of the two heavy chains. So the light chain doesn't come down that, okay, sorry. Light chain is not in the FC region. Okay, it doesn't make its way down here. Only the two heavy chains end up down here at the FC region. Good. Following along, got that? It's held together by disulfide bonding. That's key to remember. And you're like, chemistry. What? Disulfide bonding, right? So you can see these disulfides right here, sulfur groups, and they bind to each other to hold the immunoglobulin together in its shape, form. We're going to see that that's kind of not really the dimension that it actually looks like, but it's close. And we'll see a real picture of what an immunoglobulin looks as it binds around and shapes itself into this shape that we draw out. So one-dimensional is the way I call it. Okay, so an immunoglobulin is not one-dimensional. So this in FC region, what you need to know is it's important in the effect function of the immunoglobulin molecule, opsonization and complement fixation. Now, complement fixation, we just said, it's coming later. You'll catch up to this name, but you got to know it now. And I think complement's in this next unit, so this will make sense in the unit. Opsonization is what? What is that again?
Like effectiveness in the immune system. How do I say it?
Makes it what?
I had like 3 words.
Makes it taste better, right? For the phagocytic cells. So it's that immunoglobulin is all over the antigen, right? It's around, right? It's attached, right? Opsonization, it's easier for the phagocytic cell to either catch the antigen, maybe it's already caught. Maybe this end, oops, let's do this. I'm gonna keep coming back, but we will. Maybe the antigen's caught here, right? So opsonization of that antigen would be increased because it tastes better here with FC binding to the phagocytic cell is able to bind to the FC region. So it helps capture the antigen, get it closer to the phagocytic cell so it can eat it. Okay? That's opsonization. You want to think of it as flavoring, you want to think of it as increasing the attractiveness of an antigen. That's what it is. The Fab region, we're up here. Okay? So they call it in between the two arms of the immunoglobulin. But it has one light chain, okay, and 1/2 of the H chain. So this is a Fab region by itself. This is a Fab region by itself. Okay, so the top part where it says one light chain and 1/2 of the heavy chain, this is 1/2 of the heavy chain starting right here. So what they're showing you here is where we can divide the immunoglobulin. We can break it apart. Obtained by papain digestion of the immunoglobulin. It breaks the Fab from the FC. It is a specific enzyme cutter. It cuts the immunoglobulin where it wants. And the thing is this comes very important in the next step of next semester, which is your blood bank, immunohematology. This is how we go about hammering immunoglobulins a patient may have, okay? Patient has developed an immunoglobulin against an antigen, and we want to get rid of that off the red cells. We treat the red cells with papain, okay? And it cleaves off what? It cleaves off the Fab region. Can't really do much if you don't have the Fab region, right? And that's how you treat red cells. So you're going to hear that again, and that's why we have this class in front of immunohematology. It's because when Dr. Walls throws that out there, y'all aren't going, well, we have no idea what you're talking about. What do you mean papain does that? And now you know, right? So we introduce little things here and there in this chapter that you will use for blood bank. Okay? And all this was in the discovery. All of this was used to discover the immunoglobulin and to find out its structure. Now we're using it to treat patients. So if you put Fab times 2, right? This is another one here, right? We use pepsin, can do that. And what pepsin does is actually digest the FC region. So the FC region like disappears, okay? But we're able to hold on to the Fab region. But the FC portion is digested by pepsin treatment. So this is all, you know, what did we do to discover these things? And then even in 1918, well, we go back to 1845, sorry, way back, we got Dr. Henry Bench Jones. And if you hadn't heard of Bench Jones proteins, if you have, there's a specific problem. Another question for my readers ahead of class. And I know you just came off a test and you're like, we had no desire to read your chapter before we started today. Right? Y'all can all just say yes, that's us, right? All right. So Henry Bench proteins, they're found in the urine. Because you're gonna guess what, guys? You're gonna see Bench Jones proteins everywhere. When you get to body fluids and urines, you're gonna see it again later in chemistry. We're gonna use it to detect these proteins in the urine. All right, in chemistry, you're going to hear Bench Jones proteins, but what is the condition that causes it? Multiple myeloma. Okay, multiple myeloma patient starts spitting out, right? Malignant plasma cells is basically what they're called. And they find these protein, this overwhelming amount of protein ends up in the urine. Okay, so there's this new protein. You do a urine test. Y'all started urine testing? Y'all in nowhere? Okay, one of the things with urine testing is to check the kidneys for happiness, right? And if your kidneys are happy, they're not spilling protein. So all of a sudden you do a urine test and you're like, ah, this patient's sick. Let me dip my dipstick in there and wow, look at the protein indicator, blows up. We got 4+ protein in a urine, could be indicative of a multiple myeloma, okay, because these Vince-Jones proteins are there. Okay, so this was discovered in 1845, okay, and basically what they did was they analyzed the protein, they heated them up, and they revealed that we have kappa chains and lambda chains. Okay, so there's two types of chains in the immunoglobulin. There's kappa and lambda, and each one of these contain 200 to 220 amino acids. And you're wondering why you went into biochemistry. You know in biochemistry yet? No, not yet. You know what an amino acid is? Building block of a protein. Easy enough, right? So the amino acid sequence gives us our protein. And what we ended up finding was the kappa and lambda made up the constant and variable regions of the immunoglobulin. Okay, so we're going to come back to kappa and lambda when we talk about how the actual immunoglobulin is coded for and developed. So keep that in mind. So we're just introducing the kappa and lambda here, okay? The heavy chain, all right, variable region, first approximately 110 amino acids. Okay, so the whole heavy chain has a variable region and has a constant region. Don't lose that. Don't lose that idea. Don't think that the constant region is only the bottom part of the immunoglobulin. Okay, it does extend all the way up, extends up into the arms. So we have 110, we have a constant region of the remaining, and then there are 3 or more regions with very similar sequence designated as constant heavy 1, constant heavy 2, constant heavy 3, and we've already seen, go back, right? Constant heavy 1, right here. Constant heavy 2, constant heavy 3. So that's where we are now. So we're just basically talking the development. Now we have heavy chain sequencing. Heavy chains are unique to each class, and it gives each class its name. Okay? So we have what is known as a gamma chain for G. We have a mu chain for M, alpha chain for A, delta chain for D, and epsilon for E. So we have our Greek alphabet being used. It's the identifier for the chain, the different chain for each class is how they got their name, okay? So they identified those chains with the alphabet, and thus they came up with their IgG. So immunoglobulin G, immunoglobulin M, immunoglobulin A, immunoglobulin D. But this is good. You need to know this difference between these 3 terms. Okay, we got isotype, allotype, and idiotype. And you will have to know this. This will pop back up on you. So an isotype is simply a unique amino acid sequence that is common to all immunoglobulin molecules of a given class and a given species. Okay, so all of our IgG, okay, parts of our IgG are the same. All right, there's part of it, my IgG, there's a part of your IgG, parts of everybody's IgG. Since we are the same species, our IgGs are identical, parts of them, okay? That's the isotype, common to all in the species. Allotypes are minor variations of the isotype sequence that are present in some individuals, but not others. So our isotype is the common part. The allotype is, there's a tweak. There's a portion that my IgG is different than your IgG. Okay, we're the same species, but we have differences, and an allotype. And then there's idiotypes, which are variations in the variable region that give individual antibody molecules their specificity. Okay, so that's the big change. The variable region has idiotype, which is there are parts of that that are variations, so we have a unique antibody. Maybe I haven't been exposed to the antigen you got exposed to. So you have a different idiotype because you have been exposed to a different antigen. Okay? So you have a unique part of your variable region that I don't have. But there's part of our IgG that's unique, same for both of us. Then there's slight variations, the allotypes that could be talked about for some but not others. And then the idiotypes are unique to the antigen exposure. There's a hinge region called the H chain for hinge, okay, and it is located between the CH1 and the CH2 regions. Where is that? All right. Right here is the hinge region between the CH1 and the CH2. This is the hinge. Oops, went all the way back, so I'm going the wrong way. Has a high content of proline, allowing flexibility, and hydrophobic residue, so water-fearing residue. So there is a flexing of the immunoglobulin. And what it allows is two antigen binding sites to operate independently. So if it couldn't get, if you couldn't get the arms to separate or to move or to flex when they grabbed a hold of an antigen, maybe they could only grab one, right? And you would block the other one. Well, the immunoglobulin can kind of flex around and maybe do this, right? Maybe bend out where it could reach this antigen, but it doesn't interfere with the binding over here. So what we call the binding site of the Fab, an IgG can bind two antigens. It has unique binding. I can bind here and bind here. And we're going to see when we get to the IgM, it's got 5 IgGs, basically, what you look at, all right? And it can bind 10. So when we do a lot of our testing, we like to use IgM because it binds better, because it has more binding sites. Just that simple. And of course, we talked about the complement cascade again here at the bottom, coming later. Preview. So what about this carbohydrate portion? Remember, we're glycoprotein. So we have a carbohydrate found in all types of immunoglobulins, localized between the constant heavy 2 and the two heavy chains. It increases its solubility. It provides protection against degradation and enhances the functionality of the FC domain. And I know that list is like, really? You want me to remember that? Well, it is a list. It is on a PowerPoint. It is the reason we have the carbohydrate region. So yes, solubility, protection, function of the FC. And this is what an antibody actually looks like in a 3-dimensional shape, okay? So it folds amongst itself. There's immunoglobulin folding. Okay, and we have something called CDR. Does anybody know what the CDR stands for? Because I hate these abbreviations when I don't know what they mean. It's just something we need to know, right? Found on page 65. Yes, complementary determining region. Okay, great. It is, so the 3 small hypervariable regions consisting of approximately 30 amino acid residues are found within the variable regions of both the heavy and the light chain. And each of these regions is called a complementary determining region. Okay, so this is the variable domain. This is the variable of the heavy chain. This is the variable of the light chain. Attached to what? The constant domain. So now we get to the good part. Who is who? Who's our IgG? These are the things we have to associate with each one. So this is a list. It's in a table. You get a table on page 66 that divides them all up and kind of gives you a table form of these. But the predominant immunoglobulin in the human is the IgG, making up close to 80% of the total immunoglobulin in the serum. It has the longest half-life because we like that. We need it. We need IgG to hang around a little longer. Okay, 23 days is the half-life. So if you don't know what half-life means, in 23 days, half the IgG that was there is gone. The other half is about to be gone. So 23 is kind of the average, length of days that an IgG hangs around. We have subclasses of IgG. We have 1, 2, 3, and 4. You see IgG1 is the predominant one here. 66% of all IgGs are Ig1. Ig2 is 23. Ig3, 7, and 4 for 4. And the difference in the number and position of disulfide bridges between these of the gamma chain determines which class we have. So you have a picture of that on 66. I don't know if there it is. Now these are the differences. So again, the difference in the class of IgGs is based on the number of disulfide bridges. You want to call them the ladder rungs, whatever you want to call them. But IgG1 has 2, IgG2 has 4, IgG4 has 2, and then IgG3, it's got a whole ladder there. It has what, a dozen? Like that. You can see the difference in the shape, the length of the actual IgG as the classes. Since IgG3 has the largest Hen region, it has the largest number of interchain disulfide bonds, it becomes the most efficient IgG for binding complement. It's induced in response to protein antigens. So if I ask you on an exam which IgG subclass is the most efficient in binding complement and listed IgG 1, 2, 3, and 4, you need to remember that 3 is. And when we get to complement, we'll kind of see. But complement kicks off with binding of the immunoglobulins, either G or M. IgG2 and IgG4 have shorter hinge segments and they're poor mediators of complement, but they're involved in responses to the polysaccharide antigens. So which one binds protein antigens? And antigens can be any form, right? Certain antigens are better than others. And I think lipid antigens aren't very antigenic, but protein antigens would be, polysaccharides would be. So what is the function of the IgG? To provide immunity for the newborn. Because why? Because the IgG class is the only class that can move across the placenta barrier. So, blood bank story to come. We have hemolytic disease of the newborn because the IgG from mom moves across. Y'all heard this story, hopefully? No? Okay. I don't know if we have time to tell it today, but we will tell it. The IgG crosses over. So that provides immunity for the baby, right? Mom's IgGs go across, and IgGs love to get antigens, right? They love that. But sometimes that IgG can go across and cause harm to the baby, and that's what hemolytic disease of the newborn does. And this is why if you're an Rh negative mom, you have an Rh positive baby, there's problems. Okay, that antibody can be to D of the red blood cell, the D antigen, the Rh antigen. So we have that. So that's key to the IgG. That's the one thing you have to know. You have to know it's the one that crosses the placenta. It's great at fixing complement. We've already talked about that enough, I think, without knowing what complement does. Coats the antigen for enhanced phagocytosis, which is opsonization. It flavors the antigen. Neutralizes toxins and viruses. So this is key to why we need it floating around and participating in agglutination, which would be a precipitation reaction. Okay, so we can do that when we do agglutination, where we find something, capture it, and precipitate it out. Okay, so we can get rid of things with antibody binding to the antigen. So all the subclasses of IgG can cross the placenta. Macrophages, monocytes, and neutrophils have an Fc receptor to the IgG. So we've already talked about that part of the IgG binding to the macrophage, monocyte, or neutrophils, and that increases the efficiency of phagocytosis. A high diffusion coefficient allows IgG to enter extravascular spaces. So it's not in the vascular system, it actually gets out, okay, and gets into some extravascular spaces. So it plays a major role in neutralizing toxins and viruses by getting out, right? So these toxins may be in spots of your body that we need the IgG out there to pick up on. Questions right now? Yes.
This might be a dumb question.
Never a dumb question.
Is IgG also the one that's found in the mom's breast milk, or is that a different one?
There's one more in particular for that. We're getting to it. But it's a great question. Yeah. We have one coming up that likes to be in secretions, and that's when it ends up in mom's breast milk. But you are correct, there is one in breast milk. So we use IgG for agglutination, precipitation reactions, and in vitro. That's what we do in lab. And IgG is better at precipitating reactions than at agglutination. And as you would probably wonder, IgM is better at agglutination. Okay? So we'll spend some time on IgM. It looks like this lecture is going to bleed over into next week. So this is one of those that's going to force us to spend a little more time. Because this is good stuff. I don't want to leave out anybody before we move on to immunoglobulins. This is basically like the title of the class, right? Immunoglobulin. These are what we're here for, to learn about. IgM is a big one, okay? It's a big one. It's a macromolecule or macroglobulin. It's got the highest molecular weight at 900,000 Daltons. It only makes up 5 to 10% of the total. So we've got what? IgG is way out there at 80. Everybody else is going to be in the 20% total from the IgG. Half-life, very short compared to IgG, 6 days for IgM. How many for IgG? 23. Okay, great. And it can exist, and this is what you learned from the last chapter we talked about back in the first unit, right? That it can be just one by itself on a B cell. That's an IgM, okay? So when we talk about the pentamer, about the whole IgM and this big 900,000 Dalton thing, each one of those can be by itself as a monomer. So don't forget that. Don't think IgM has to be in the pentamer form at all times. But it's a pentamer, okay? And this is like a nightmare for me right here, because we were in the Reynolds building many years ago, and I had my little quiz bowl team. We were fighting for the championship, okay? Going up against the A-State team, right? And it came down to this question. The IgM, immunoglobulin, is known to be in what? Geometrical shape. Yeah? So that's like a softball, right? We've had this in class. Like, there's the softball, hit it out, win the whole competition. Be a champion for the day. So the nightmare was, my captain goes, got it, know it. And he says, heptin, or heptagon. Where did heptagon come from? When have we ever said when have we ever said 6, right? It's always been 5. Oh, it never has been 6, it's been pentamer. So that's the nightmare of this lecture right here for me. It's a flashback into losing the Quiz Bowl championship. That still hasn't grasped that champion's trophy yet. And pentamer was the reason I didn't have that glory one day. So hopefully you don't forget pentamer. But would he have been correct if he said monomer? I would argue yes. He could have said monomer, or I don't know if that's is monomer a geometrical shape with maybe one? I mean, I know it means 1, but I don't know. He would have probably had to argue that one. We probably had to fight over that one. So there it is. So there's our big IgM, and that's where we're going to leave it. We're going to pick up with IgM on Monday. Okay, but right now that's the shape. So you look at it, it looks like 5 IgGs put together, but a little length in there, right? The IgG doesn't always have that long of a heavy chain. But that's the pentamer story. You'll have a wonderful weekend. Okay, be on the lookout for micro. Be on the lookout for the review quiz pop up this afternoon, hopefully before noon. I'm going down there to work on it right now, and I'll make sure I got it good before I post it and let you start working on the micro 1, unit 1 review. All right. See y'all later, Zoomland.